concentrate on atomic structure, chemical bonding, water properties, and also carbohydrates. Right. So the first question we have here, when a molecule loses a hydrogen atom, that molecule is said to be oxidized, reduced, hydrolyzed, hydrated, protonated. So when you're talking about loss of hydrogen, gain of oxygen, loss of electrons, that's oxidation. So that atom is oxidized. The other way around, the loss of oxygen, gain of hydrogen, or gain of electrons, that is reduction. Okay, so that atom is reduced. Now, hydrolysis, or to be hydrolyzed, is to be broken when you react something with water. For example, when you react maltose with water, it's going to be broken down into two glucose monomers. Now, hydration, or to be hydrated, is to gain water. To be protonated is to be reacted with a proton, that is hydrogen ion, or to gain an hydrogen. All right, so question number 2. Chemical properties of atoms are determined by protons, electrons, both protons and electrons, nucleus and neutrons. When you're talking about chemical properties have to do with the reaction of that atom. So the chemical properties or the reaction of an atom is determined by the number of electrons. If we are talking about the physical properties, that will be determined by the number of protons and also neutrons. Question 3. An atom has a mass number of 23 and atomic number 11. The number of protons are, so this is just direct. If it has got atomic number of 11, meaning that the number of protons is going to be 11, because the atomic number is determined by the number of protons. Question 4. The S and P shells of an atom are full. Its atomic number is. So when you're talking about filling up the shells, we are going to follow some rules. That is the Hans rule, the Aufbau's principle, the Pauli's exclusion principle. So we've got first the 1s orbital, we've got the 2s orbital, we also have the 2p orbital, and then we have got the 3s orbital, we have got the 3p orbital and the 3d orbital. We'll continue going like that. So when we are filling up these, we always start first with the 1s orbital to fill. So if you fill the 1s orbital, you are going to fill the 2s orbital, and then you are going to fill the 2p orbital, and then you can fill the 3s orbital like that. So the s orbital always occupies a maximum of 2 electrons, the p a maximum of 6 electrons. So first you fill the 1s orbital with 2 electrons. The next one you're going to fill is the 2s orbital, also with 2 electrons. From there now you're supposed to fill the what? The 2p orbital with a maximum of 6 electrons, because the question is saying the s and p orbitals are all full. So if you fill the 2p, you're supposed to fill the what? The 3s. Okay, the 3s orbital with also 2 electrons. After filling the 3s, you're supposed to come and fill the 3p, also with 6 electrons like that, and then you continue filling. Now we're only interested in the s and the p. So if we fill the first 1s, 2s, and 2p, this is going to give us 10 electrons. We will fill the s and the p. Or if we fill the 1s, the 2s, the 2p, and the 3s, that is going to give us 12 electrons. If we fill everything here, 1s, 2s, 2p, 3s, and 3p, that is going to be 18 electrons. So from these which are here, which one is present on the options? So I'm able to see that is 10. So that is 10 electrons. It can continue going, we can continue going like that, but if we continue going and going, of course we are going to come and fill the 4s there. Okay, now as you get to fill these other things here, you will also require to fill some other orbitals like the 3d and these others. Let's consider question 5. Which of the following is correct about the carbon 12 atom? Now, carbon 12 has got 6 electrons, it has got 6 protons, it also has 6 neutrons. That is because its mass number is 12. And we know that carbon 12 has got 6 protons. So if something has got 6 protons, its mass number is 12. Of course, we know that the number of protons is equal to the number of electrons. And to find the number of neutrons, it should just be mass number minus number of protons, which is 6 still. So the answer is 1, which is 666. Question 6. An element has an electronic configuration of 2,8, 7. Its valency is. Now, valency is just a combining power. So you look at the electron in the outermost, the outermost shell. It has got 6 electrons. So it requires 2 more electrons for it to reach an octet. Okay, so the valency is 2 for that one. Question number 7. Atoms are electrically neutral under normal circumstances because they have equal number of charged and uncharged particles. Now, it shouldn't be charged and uncharged. That will not be electrically neutral, because something that is electrically neutral should have positive and negative things balancing up. Have neutrons in their nuclei. All atoms have got electrons in their nuclei. Even equal number of negatively and positive charged particles. This is true. Have an equal number of neutrons and protons. If it was electrons and protons, it would have been making sense and it would have been correct. But neutrons are neutral, protons are positive, so this can't be electrically neutral. If more neutrons than electrons, this is also not true. So we've seen the answer there is a 3. Question 8. Which of the following statements about positive ions is correct? They are also known as cations. Yes, we know positive ions are also known as cations. This is true. They are formed when electrons are removed from an atom. Yes, when an atom loses, okay, or gets removed some electrons, it is going to become positively charged. They are smaller than the atoms from which they are formed. This is also true. The reason is this: think of an atom that has got, let's say it has got, the protons are 6 and then the electrons are also 6. We know that a neutral atom, the number of protons and the number of neutrons are the same, meaning that in the nucleus of this atom we have got positive things are 6, and then now the electrons are around in the shells, so we have got negative 6. So these are all balanced. The protons and the neutrons are balanced. Now if you lose electrons, for example you lose, let's say 2, you are going to remain with 4 of them. The proton number doesn't change. So in this case, what you're going to see is the nucleus is still having a positive 6 charge, but the shells, the charge is now what? Negative 4. So what you are going to see is there is an attraction between positive and negative things. So these electrons are going to be more attracted, because what is attracting the electrons is more than what was attracting electrons in here. Okay, you think of this. You have 6 people pulling 6 people. The force is not going to be that great as 6 people pulling 4 people. So because of this shell is going to be more inside. It will be pulled more inside like that. That is why it ends up becoming what? Becoming much smaller. So cations are smaller than their neutral atoms. Okay. Option 4, they attract negative ions. We know that positive things attract negative things. This is also true. So the best option here is all of the above. So this is going to be the answer then. Question 9. Which of the following statements about sodium chlorides is correct? It is soluble in water. We know that is true because sodium chloride dissolves in water. It has a melting point. Obviously, because this is a solid, there is a point at which it is able to melt when you get to expose it to heat. It is brittle. Yes, it is brittle. Brittle means it's able to break. You can think of this. Sodium chloride is table salt. Table salt is able to break like glass. It conducts electricity at room temperature. Now, sodium chloride can't conduct electricity at room temperature unless when it is in molten state. You can think of it, if you get salt just the way it is, you pass electricity through it, it will not pass until you get to melt it. So the answer is all of the above options are correct except 4. So this would be the best option. Van der Waals forces are ionic bond interactions, hydrogen bonds, covalent bonds, sudden dipole-induced forces, hydrophobic interactions. So when you're talking about Van der Waals forces, also known as London dispersion forces, these are just sudden dipole-induced forces. Okay. Question number 11. Which of the following is not true of water? In ice, water is bonded to other four. This is true. Okay, in ice, each water molecule in ice is bonded to four other water molecules. That is why it has got a solid shape. Water molecules have a melting point due to intermolecular hydrogen bonds. This is also true because of hydrogen bonds between different water molecules. That is why water has got a high melting point. Water has intermolecular adhesive forces. This is also true because water gets attracted to other surfaces that are not water. Water has a lot of hydroxyl groups. This is also okay. This is also true. It has got a lot of OHs. If you look at water, there's an OH inside there. Okay, the way water is like this. So this is an hydroxyl group. Water is solid at room temperature. Now this is not true. Water is not solid at room temperature. It is liquid. So that is the answer. That is not true. The valence electrons of representative elements are the valence electrons of representative elements are the ones that are of chemical importance. So valence electrons are electrons in the outermost shell, and these are the ones which determine how an atom is going to react. So they are the ones of chemical importance. Are in s orbitals? No, this is not true. Valence electrons can be in any orbital as long as that orbital is in the outermost shell. So it can be in s orbital, p orbital. That means that even this one is not going to be true. Located closest to the nucleus, this is not true. Valence electrons are in the outermost shell. In p orbitals located closest to the nucleus, this is also not true. So it's in the best answer is 1. Question 13. Polar refers to bonds that have an even distribution of charge. This is not true. When you're talking about polarity, polarity is going to be due to uneven distribution. For example, if you've got hydrogen is bonded to oxygen, what you are going to see is because oxygen is more electronegative. If you want to tell which atoms are more electronegative, check on the periodic table. Here is fluorine at this point. So fluorine is the most electronegative atom. So anything that is closer to fluorine is going to be more electronegative. So electronegativity increases as you go towards fluorine like that on the periodic table. And we know oxygen is more nearer to fluorine. So electronegativity is just the ability of an atom to put electrons in a bond. So because of this, when you see one bond like this, it means there are two electrons. So oxygen tends to put electrons more to itself, but it doesn't take them. It is just more like it is putting them more closer to itself, but hydrogen is also having a hold on them. Now we know that electrons are negatively charged, and because the electrons are more closer to oxygen, this is going to give oxygen a partial negative charge, and hydrogen gains a partial positive charge. So this partial negative and partial positive is what leads to what we call polarity, polar bonds. So polar has to do with uneven or unequal sharing or distribution of electrons. So the formation of an even size ion, this is not true. Bonds that have uneven distribution of electrons, this is true. Atoms with the same electronegativity. Now if atoms have got the same electronegativity, they are going to have the same force to pull electrons, and so electrons are going to be just on the middle. They are going to be equally shared. Double bonds in a molecule, that is also not true. Which kind of bonding can be found in a sample of water? So because water contains oxygen and hydrogen like that, so expect that water is going to have, okay, it is going to have a polar bond. Now this polar bond is covalent because it has to do with the sharing of electrons, and in water, because water molecules tend to pull each other, there are also hydrogen bonds. So in water you find polar, covalent and hydrogen bonds. Okay, when an ionic compound is dissolved in water, what happens? Ions dissociate in water. This is true. Okay, ions dissociate in water. Think of sodium chloride. When you've got sodium chloride, this is going to dissociate in water, and what you're going to see is the sodium ion, the negative parts of water, that is what the oxygens are going to be like this. These are going to be the hydrogen. They are going to surround this oxygen in this manner. And then the chlorine, which is negative, the positive part of water, that is the hydrogen, also surrounds the chlorine in that also manner. So this is what happens for something to dissolve. Okay, ions dissociate in water. The compound gets hydrolyzed. Now, to be hydrolyzed is to be broken when you react something with water. This is not true. Ionic compounds are not broken because they react with water. They only break because they disassociate in water. Okay, hydrolysis, you can describe it when you're talking about, maybe let's say, maltose. If you're talking about disaccharides, polysaccharides, those which lose water when they're being formed, if you react them with water, they're going to break. Okay, you can think of esters which lose water when they're forming. So when you react an ester with water, it's going to break. That is called hydrolysis. So the compound gets hydrated. Hydrated means gaining water. Okay, gaining water inside it, and that is not really true. Okay, the gaining of water you can think of when you get to vomit, you lose water. So hydration, you get to now take in the water, get to drink that water. Ions get surrounded by water. This is what you're able to see for an ionic compound. Ions are going to be surrounded by water. So this is also, this option is also true. So meaning that 1 and 4 are what? Are correct. Okay, that's the answer. Question 16. Starch is made from. Now, when you look at starch, starch is first going to come from glucose. And these are alpha glucose molecules. They are going to form first maltose, and then maltose now forms the starch. Okay, so it comes from alpha glucose monomer units. The sugar present in milk is called, well, milk sugar is lactose. We need to know that. Question 18. Which of the following is not correct about water? Water molecules are cohesive. This we know is correct. They are cohesive, meaning that they attract each other. Adhesive and cohesive forces of water molecules account for capillary action. This is true. The reason as to why water is able to move in a vessel, you can think of the xylem and the phloem. The reason as to why water moves down going up without any force pushing it is because of cohesion and adhesion. The density of water is greatest as 4 degrees Celsius. This is also true. Water is densest as 4 degrees Celsius. Cohesive forces are responsible for wetting surfaces. This is not true. Cohesion has to do with one water molecule attracting the other water molecule. So the reason as to why surfaces wet or our clothes wet when we spill water on them is because of adhesion. Water is attracted to other surfaces. Water helps mammals to maintain a stable body temperature. This is also true. So the one which is not correct is option 4. 19. The beta 1,4 glycosidic bond between two glucose molecules was resulting the formation of number 1, sucrose. Now this is not going to be true because sucrose is made from beta fructose and alpha glucose. If you talk of cellulose, this is going to be true because cellulose is made from beta 1,4 glycosidic bonds. That is from beta glucose molecules. Maltose is made from alpha 1,4 glycosidic bond, not beta. And lactose is made from beta galactose and either beta or alpha glucose. So this is not going to be true. Okay. Amylopectin is starch which is made from alpha 1,4 and 1,6 glycosidic bonds. So this is not also true. Question number 20. Which carbon gives an hydroxide and hydrogen to form the glycosidic bond in hydrogen? This is supposed to be in maltose. So this molecule which we've shown here is maltose. So which one is for this bond which you are able to see here, which one is going to donate an hydroxide and which one is going to donate an hydrogen? So if we look at this one, I'm just going to pick this point here. We're able to see what you are going to see is you've got H and then you have got an OH, and then you have got also this side where you've got H, you also have an OH. So this is carbon number 1 of one glucose molecule. This is carbon number 4 of the other glucose molecule. So what you're able to see is this hydrogen, which is from carbon number 1, is going to react with the hydroxide from carbon number 4. And that is going to form a water molecule that is going to be released. That is why we say these reactions are dehydration or condensation reactions. So the hydroxide comes from carbon number 4, while the hydrogen comes from carbon number 1. So that means the best option is option number 2. So hydroxide is from carbon 4, and then the hydrogen is from carbon 1. Okay, so that's the answer then. Which of the following molecules is used for the synthesis of nucleic acids? Now, if you're talking of nucleic acids, this is DNA and RNA. From the functions of carbohydrates, we said pentoses are important in the formation of DNA and RNA. You can think of ribose is important in RNA, while deoxyribose is important in DNA. Of course, we also have ribulose, which is a pentose. Now, that one gets to come in when you are talking about acceptance of carbon dioxide in the process of photosynthesis. Question 22. Identify the anomeric carbon in dihydroxyacetone. So dihydroxyacetone is a triose, which is a ketone, and the functional group is at carbon number 2. So that is where the anomeric carbon is. Anomeric carbon is just a carbon containing the functional group. Question number 23. Study the structure below and give the correct match between the structure and the name. If we look at the first one, so how many carbons does it have? 1, 2, 3, 4, 5 here, and 6, meaning that this is a hexo sugar. And look at the way the hydroxides are. The hydroxide is here, another hydroxide here, another hydroxide here. So whenever you see this kind of shape, this is glucose. Now, because the OHs are more concentrated on the left, this is going to be L-glucose. If you look at the second one here, it is also having 6 carbons. 1, 2, 3, 4, 5, and the 6th one here. But look at the way the hydroxides are. They are making more like a C. So just know that this is galactose. Now, if the OH here is on the right, this is D-galactose. And look at this one. It has got how many carbons? 1, 2, 3, 4, 5, meaning that it is a pentose. Now, if you look at the OHs, they are all on the right. So this is going to be D-ribose. Okay. At carbon number 2, if we only had an hydrogen instead of hydroxide, that would have been D-deoxyribose. So the answer is option number 3. Choose the correct statement in relation to the structure of disaccharides. Choose the correct statement in relation to the structure of disaccharides. Maltose is composed of beta and alpha glucose. We know that maltose is composed of what? Alpha glucose. Sucrose has a glycosidic bond between carbon 1 of glucose and carbon 4 of fructose. We know that sucrose, it is carbon 1 of glucose and carbon 2 of fructose which come together. Lactose is composed of alpha glucose molecules. You know that lactose is made of what? Galactose and glucose. Sucrose is composed of alpha glucose and beta fructose. This is true. It is alpha glucose beta fructose. Maltose is composed of alpha and beta fructose. This is not true. Question 25. Maltose is a non-reducing. Choose the correct statement. Maltose is a non-reducing sugar because of the 1,2 glycosidic bond. Now, the 1,2 glycosidic bond is not even in maltose. Maltose is a reducing sugar. Sucrose is a reducing sugar. Just the word reducing sugar for sucrose means it's wrong, because sucrose is a non-reducing sugar. Why? Because it has got a 1,2 glycosidic bond. Lactose is a non-reducing sugar because it is found in animal milk. This is not true. Lactose is a reducing sugar. The only non-reducing sugar for disaccharides is sucrose. So this is the best option. And why is it? Sucrose is a non-reducing sugar because of 1,2 glycosidic bond. And the reason for that is carbon number 1 of glucose is the one which contains the functional group which has got reducing properties. And carbon 2 of fructose is the one which contains the anomeric carbon, or the functional group, which has got reducing properties. So you've seen that all carbons with reducing properties have reacted. That is why it tends to be a non-reducing sugar. Question number 26. Which of the following molecules is most suitable for translocation in plants? Sucrose, cellulose, glycogen, starch, galactose. while sucrose is very best for translocation in plants. Milk sugar is made from maltose and fructose, glucose and fructose, galactose and glucose, galactose and galactose, glucose and glucose. Now, milk sugar is simply lactose, and lactose is made from galactose and glucose. 28. Choose the correct statement. Branching is important in the formation of structural polysaccharides. Is it? So we have got two types of polysaccharides. We've got those which are involved in structures, which we call structural polysaccharides, and then we also have those for energy storage. Now, for energy storage, because we need energy anytime, they should be branched. Okay, the reason as to why they should be branched is so that they can be easily broken down. But the structural polysaccharides shouldn't be branched. They should be unbranched, because think of the cell membrane. You need something to surround the cell like that. Okay, so it shouldn't be branched. So branching is important in the formation of structural polysaccharides. That's not true. And energy storage polysaccharides should be fibrous in nature. Energy storage shouldn't be fibrous. It's those which are used in structures which should be fibrous, as I've drawn here. Okay, fibrous means they should be long with fibers like that. Amylopectin is highly branched and therefore more difficult to digest than cellulose. Now, amylopectin is of course a starch. It is branched, yes, and because of being branched, that is why it is more easy to digest, because it can easily be broken down. Remember, amylose is there for what? For storage of energy. I mean, amylopectin is there for storage, just like amylose, which are all parts of starch. But cellulose is used in structures, it's found in the cell membrane. That is why it is difficult to digest, but this option is saying the other way around. Amylopectin is highly branched and therefore more difficult. It is actually easy to digest. The structure of glycogen is similar to that of amylopectin. This is true. Glycogen is branched, it has got few branches, and amylopectin is also branched, it has also some few branches. So there are alpha 1,4 and alpha 1,6 in both amylopectin and glycogen. Amylose gives a blue-black color with iodine solution. This is also true. Okay, this is also true. When you get amylose and you get to treat it with iodine solution, it's going to tend to blue-black. That is because the amylose gets to interact with the what? With the iodine solution, and this is what gives the taste for starch, blue-black color. Now, which one would you choose? Because both 4 and 5 are correct. So that is why you need to get to choose the best option. And the best option we can really go for is this one. Of course, the structure of glycogen and the amylopectin, they are that similar, but they are not the same. So this would be a very good debate to debate on, but the best one we can go for is there is no other option. When you get amylose with iodine solution, it will always give you blue-black color. Okay, but glycogen and amylopectin, though they've got similar bonds, they are not really that similar. They have got similar bonds, but not really that similar. Okay, choose the correct statement. Amylose has alpha 1,4 and beta 1,4 glycosidic bonds. This is not true because we don't have any beta 1,4 glycosidic bonds in amylose. We only have alpha 1,4, that's all. Amylopectin has alpha 1,4 and beta 1,6. This is also not true. Amylopectin has only alpha 1,4 and alpha 1,6. Okay, not beta. Cellulose has alpha 1,4 and beta 1,4. This is also not true. That is because in cellulose we only have beta 1,4. Glycogen has alpha 1,4 and alpha 1,6. This is true, as we said, glycogen and amylopectin have got similar bonds, though they are necessarily not similar, but they've got similar bonds. Glycogen is a storage form of energy in animals, while amylopectin is a storage form in plants. Amylose has alpha 1,6 glycosidic bonds. This is not true because amylose has got alpha 1,4, not alpha 1,6. The complete hydrolysis of amylopectin yields amylose, galactose, glucose, fructose, starch. So amylopectin is a starch, and the starch is made from maltose. Maltose is made of glucose. So when you get to hydrolyze, or when you get to react amylopectin with water, you are going to form glucose at the end of it all. Well, thank you so much for watching up to the end. Hope that was